Welcome back to another video. Today I'm going to be talking about plant transformation and the use of agrobacterium in plants. Here we go. So first of all, the use of agrobacterium. They are the natural engineers, so they occur naturally. In the wild, agrobacterium bind to the wounded sites on plants. This is how they gain entry. Obviously, they can't go through the bark or the waxy cuticle of plants too thick. Then they send their genetic information into the cells, into the nucleus, and then it will join into their DNA. This then tells the plant to start making food for the agrobacterium, such as sugars, and it will also, the DNA that the agrobacterium holds will also encode and force the tree to start to perforate tumors and making the cells with the new DNA grow and replicate excessively. So if you walk around some parks, you'll see this almost lumps of bark on older trees, oak trees. This is all from agrobacterium infections. So agrobacterium can be used to make GM crop. You insert your desired gene into GM crop as genetically modified crop. You insert your desired gene into the plasmid of the agrobacterium. Insert an antibody resistant gene. This will be useful and we'll talk about it later on. And remove the section of the gene that promotes tumors. Obviously, you don't want your plant just growing tumors once you've made it resistant to and inserted your desired gene. Then you add it to a wounded plant, as it would happen and occur in nature. Obviously, you can wound the plant yourself. The DNA will go into the genome, so it will integrate itself with the plant's DNA and then you can select the cells you think may have taken up the gene. So obviously you're not going to be able to know until you test for it. So you just take a selection. Then you add these cells to a selection medium. So any cells that do not pick up the inserted DNA from the plasmid will then die. So all the DNA. So you've added a antibody resistant gene into the same section of DNA that you have your desired gene. So if any of the plants haven't picked up the desired gene, they also wouldn't have picked up the antibody resistance gene. So they will die when you add them to the selection medium. These cells have picked up the DNA will now be able to be isolated. So you've tested which ones they are, you isolate them into a separate petri dish, and you add them to a growth medium with nutrients, etc., to grow the new plant. So you've grown it now from one cell. So when the plant grows, every single cell will have this new resistance or the desired gene that you added, for example, of resistance to a virus. And this whole process can be completed within only a few months. However, some plants aren't susceptible to agrobacterium. They have grown resistance, or the agrobacterium just can't get in. So if this is the case, then you can fire a particle gun with the desired DNA directly into the plant, and then you can use the process of selection and regrowing, similar to the section here where we can just add it to a selection medium, and the cells that did not pick up the desired plasmid, the desired gene or the desired resistance gene will die, and then again you can regrow it using a medium growth. Hope you enjoyed this video. Thank you very much.